Millions of people are impacted annually by musculoskeletal tissue damage caused by various factors, including fractures or old age. Restoring their functionality is challenging due to the complex nature of these tissues. Clinical standards relying on grafts and implants have limitations, including supply constraints and high failure rates.
The reconstruction of musculoskeletal tissues has been extensively investigated due to the growth of trauma injuries and degenerative diseases that affects millions worldwide.
InterLynk, an EU-funded research project coordinated by the University of Aveiro, unites 8 partner institutions from 4 European countries. Together, they are developing innovative solutions based on advanced medical scaffolds produced with multiple biomaterials, cutting-edge virtual modeling and additive manufacturing technologies.
InterLynk is a highly ambitious project aiming to develop multi-biomaterials constructs that could be used then for the repair of maxillomandibular defects, including the temporomandibular joint.
InterLynk collaborates with Charité to draw on their extensive knowledge on the regeneration of musculoskeletal tissues, in particular the reconstruction of jaw bones and adjacent articulations that find application in maxillofacial surgery.
Many patients can have some kind of disease, some kind of tumor in their lower jaw or mandible. And in the InterLynk project, we are trying to conceptualize and build up material that can replace the need for the use of what is called grafts. Grafts are pieces of bone that we take from other parts of the body to replace the bone that is lost due to tumor or due to the dying of the bone in the area of the jaw.
Musculoskeletal systems are highly complex biological architectures consisting of hard and soft tissues, cartilage, ligament, tendon, bone and muscle. Each of them accounts for distinct regeneration processes. In InterLynk, different biomaterials will be combined to develop scaffolds that successfully respond to the diverse regenerative needs of these tissues.
The choice of biomaterials is unique and considers the different tissues and interfaces existing in such anatomical sites. It includes products based on modified fractions of human blood that could be transformed into hydrogels with high biochemical affinity to human tissues by the action of light, calcium phosphate nanoparticles mimicking the inorganic component of bone, polymers able to be processed into nanofibers to reinforce the structure, and drug carriers to stimulate tissue regeneration.
As particularly at the University of Aveiro, we are trying to develop inks for 3D printing applications. These inks will have different components in their composition and will target from stiffer to softer tissues. So, our responsibility in the University of Aveiro is to develop these inks comprising different materials.
Within the InterLynk project, a remarkable combination of biomaterials is being explored, harnessing the potential of modified fractions of human blood. These fractions, known as platelet lysates, hold incredible regenerative properties, stimulating wound healing and tissue regeneration.
In simple terms, platelet lysates are an assortment of proteins taken from the blood, and when they are used in a wound, they can stimulate the healing of that wound.
Metatissue, a spin-off of the University of Aveiro specialized in designing and developing innovative materials for cell culture, tissue engineering and regeneration, is providing the project with a series of materials based on human-sourced platelet lysates.
The main advantages in using these metatissue human-based bioinks is this high biocompatibility, has this human base and could be patient-specific, and also the possibility to control the mechanical properties, so you can match the properties of any tissue of interest that you want to regenerate or to mimic for in vitro testing.
The platelet lysate-based hydrogels can then be further combined with mechanically reinforcing materials, such as calcium phosphate nanoparticles.
The role of calcium phosphate particles is important here for the bone regeneration because they have a similar composition as the natural composition, or inorganic part composition of the natural bone.
The Dutch life science company Kuros Biosciences is a leader in the field of orthobiologics and a pioneer in the development of second-generation calcium phosphate-based products. They will supply InterLynk with osteoinductive calcium phosphate powders needed to print devices with maximum functionality.
So the challenge is to have a stable material to be able to reproduce the composition, the nanoscale, the microscale, and once it's combined by other partners with polymers, biological agents, and additive manufacturing process, we have a fully effective material that will lead to bone regeneration.
These different biomaterials will be combined to develop bioinks for 3D printing.
Each injury is different from each patient. So with InterLynk, we pretend to use the inks that we are developing to 3D print personalized implants that could be matching not only the shape of the defect, but also the properties of those tissues.
InterLynk is collaborating with the Merln Institute at Maastricht University to advance additive manufacturing technologies, cooperating in particular in the development of photocuring assisted printing and its combination with melt electrowriting and electrospinning.
So in InterLynk we try to use two advantages of 3D printing technology. The first one is that we are able to make patient-specific implants, and the second one is because we have several techniques and several materials that we can combine together, we can better mimic the complexity of the human body.
We are proposing the use of state-of-the-art manufacturing technologies to integrate the different biomaterials into patient-specific final constructs that could be then surgically implanted.
InterLynk partners with EnvisionTEC, a renowned global provider of professional 3D printing solutions, to leverage their cutting-edge 3D printing technology. EnvisionTEC's expertise will enhance the functionality of Desktop Health's 3D bioplotter, enabling the precise processing of project-developed materials and the successful printing of complex, multi-material scaffolds.
The challenge in creating that is to create a platform which doesn't limit itself to a very specific material that can be processed, but actually has a very wide range of different materials that can be processed within one single machine. So to have an all-purpose machine that many different users could use with very little adaptation by the users themselves.
INEGI, the Portuguese Institute of Science and Innovation in Mechanical and Industrial Engineering, will be using specialized software to numerically model and simulate the 3D printing of the InterLynk scaffold.
We can use numerical methods and finite element simulations to avoid the usage of animal tests, human tests, avoiding many ethical and legal problems.
Together, they are engineering complexity to shape a future where tissue regeneration is more effective and personalized.